what gets me up in the morning is actually the possibility of discovering something new. The possibility of going into the lab and doing an experiment or running some analysis and discovering something which only you knows at that given moment. So in my prize-winning research, we have tried to use the bat to provide novel insight about the neural mechanisms of the spatial representation system in the mammalian brain. And so these allowed for a comparative approach. And we targeted this comparative approach to a specific question about how grid cells in the entorhinal cortex are being generating their spatial activity pattern. And we specifically examined a very prominent class of models in the field which argued for a causal link between brain oscillations, specifically theta oscillations, and the existence of the grid formation. And what we were able to show in the bat that you can get those grid cells to exist in the absence of these very specific continuous brain oscillations, theta oscillations, and by this causally argue against the link between those two phenomena. In the second research project that I have done, we've tried to do something which was essentially impossible to do in more traditional animal models in the field, and that is to examine how the complete three-dimensional space in which we all live in every day is actually encoded by the hippocampal play cells. And for that, we developed a technology that allowed us, essentially for the first time, to record the activity of individual neurons from freely flying animals. And we targeted that, as I said, to examining hippocampal play cells. And what we have found is that these neurons in the freely flying bats encoded the three-dimensional environment in an isotropic and uniform rate coding mechanisms, meaning that the firing fields of those play cells were equally sensitive to all the different axes of the environment, and in addition, they encoded the entire environment in a uniform way, such that the animal had a complete representation of all the locations in the room, and the resolution of this representation was the same across the entire environment. I think this prize, for my research specifically, is very important because I'm using a very non-traditional animal model in my research, which is the bats. And the bats is a very unusual animal model, and I think that our work has emphasized that the use of additional mammals which can provide very interesting insights is important, and I think that this kind of a prize and this kind of an exposure to this line of research is a great benefit. So as for my plans in the future, I plan to come back, hopefully in my own lab at some point, and come back to working with the bats to study the neural basis of decision making, because I think this can be a very exciting opportunity to get into questions where really the advantages, the key advantages of the bat, such as its sensory system and its natural behavior, can really be very beneficial in studying the neural basis of decision making.
In my research, I'm interested in memory center, and in particular the memory center of the fruit fly Drosophila melanogaster, simply because it's a numerically simpler brain, so that allows us to look at connections in a statistically significant way. So what we found is that these neurons, these memory neurons, which are called the Kenyan cells, are randomly connected to the periphery. And we figured out that this randomization strategy makes actually a lot of sense for a memory center, simply because it can encode the maximum number of different combinations that a fly might encounter in its lifetime. This paper was mainly looking at olfaction, so how olfaction is, or the olfactory system is represented in these memory centers. And so right now I'm interested in looking at other senses, so like vision or touch. How are these senses then relate to this memory center, and if they use a similar strategy, or if other strategy are at play there.
My research question that I'm interested in is how memories get stored in the brain and the role that sleep plays in this process. And so for my postdoctoral research in the lab of Matt Wilson at MIT, we study a region of the brain called the hippocampus in rodents. And this area is thought to be important for memory, but also spatial navigation. So if you record neurons in this region of the brain, you'll find that different neurons like different locations in space. So as the animal is running along a spatial trajectory, one neuron might like this location, while when the rat's over here, a second neuron might like this location. So by looking at their neural activity, you could see where the animal is in space. Okay? What's really interesting is when the animal sleeps, you find the same patterns of neural activity that occurred during behavior replay. Okay? So there's a neural memory trace that's getting reactivated when the animal sleeps. So this was work that was already going on in the lab, but my question was, can we have some influence over these neural memory traces? So normally, spontaneously, these memory traces get replayed, but they can represent many different events that happen to the animal. So the question is, if you train the animal to, let's say, run to the right when you play one sound and run to the left when you play another sound, can you get a particular memory trace to be activated? And it turned out that if you use a cue to make the animal do part of the task, like running to the right, playing that cue will cause, when the animal sleeps, the memory trace to replay in the same direction. So you have some control over the memory traces that are getting reactivated by pairing a cue that was also paired with the behavioral task. What I really enjoy about my research is coming into the lab and you plug in the animal, you have wires implanted in the animal's brain, and you just put a little plug in and turn on the computer, and you can hear the activity of the brain. And it's really remarkable to hear all the pops and crackles of neurons as the animal's running around, and you can actually hear what the brain cares about. So a particular cell might like a particular location in space, so when the animal is in that location, suddenly you hear all this neural activity, and then it runs somewhere else, and then it's silent. And it's almost a mystery sometimes to try to figure out what each neuron cares about. So my future research plans is to combine my postdoctoral research studying memory processing in the hippocampus with my Ph.D. research studying perception in auditory cortex and neural coding. So understanding how the perception of sound and the memories of sounds are encoded with neural ensembles.
I think this prize is special because we are trying to reach people at an early stage in their career. We try to find people who have just started their scientific life, and we want to find who is able to do good research in the first place, but also who can communicate this research.
Frankly, I never thought I would really have a chance, and the fact of me winning it is really still a bit of a surprise to me. And especially not because I don't think the research is good, but because I think the level of the competition for this prize is so high. You look at the past winners and past finalists, and it's just daunting. I mean, the level of the competition.
So my advice for young scientists is you have to try. If you don't try, you'll never know. And just writing an essay, rephrasing your research in a way to appeal to a broader audience is a good exercise.
Sit down and write the essay and do that exercise, and not be shy, and to just submit it. Yeah.
When you're excited about your research, try to bring this across. Try to grab the attention of the judges by showing that you're really deep into this field. And it usually comes across relatively quickly in the first few sentences.
It's a lot of fun, and just the chances of just to be given the opportunity to compete in this kind of an award, this kind of a competition, I think it's a great benefit, and you should definitely go ahead and do it.